Hi everybody, welcome back to Synthetic Biology 1. Today we're going to look at where new biological parts come from. Nature is bananas. Just look at it. It's huge, it's complex. We know at least some of it is built out of simple biological parts. Things that we can use in synthetic biology to create new functions. But where are they? Where are all the parts? How do we discover new parts in nature? That's what we really want to do, right? We want to find wild organisms with completely new capabilities and then use them to do cool stuff. In synthetic biology terms, we put it this way. How do we take new functions, identify the associated genetic elements, and then introduce them into an engineered organism? Well, most of the time, you won't be out there in the field discovering new parts yourself. It takes a lot of time and work to discover a new gene, like 3 years of work, a PhD thesis level work. But fortunately, there are thousands of PhD students out there doing this and reporting their findings all the time. The scientific literature is our dispatch from the frontier. It's our #1 resource for new parts. Let's try an example to see how this works. Hey guys, check it out. I just found this really cool paper describing something called green fluorescent protein. I think it's going to be the hottest new science trend of 1992. Let's get the DNA sequence and make it into a part. Now, back in 1992, DNA sequences were rare and special enough that they actually wrote out the entire sequence in the paper. These days, you don't really see that anymore. DNA sequences are mainly shared as digital records. But if you look down at the bottom of the page, you can find the link to those digital records in the form of a GenBank accession number. GenBank is a public DNA sequence database run by the NIH. Scientific journals generally require that you deposit your sequences in GenBank before they'll publish your work. So almost all published DNA sequences end up there. So we can cruise on over to the GenBank website, punch in the accession number, and get plugged into everything that GenBank knows about this GFP sequence. Now, there's a huge amount of information here, and I encourage you to hang out and see everything that GenBank has to offer, but let's go over some key fields just to see what we've got. I see here the organism that this came from, Echoria victoria. I see the authors and the title of the publication where it was described. I see some key words here like bioluminescence that give me some clue about why this is important. I also see some information about the sequence itself. This is a 966 base pair record and it comes from an mRNA. That particular piece of information is very valuable. We expect an mRNA to have a particular structure that will help us to interpret the sequence that we see. For example, we expect an mRNA to contain a coding sequence for a protein. Take a look down here and we can find another clue. This is a cDNA sequence, meaning it was prepared by reverse transcribing RNA to DNA prior to sequencing. That means we expect to see a DNA sequence, ACTG, and not an RNA sequence, ACUG. Scrolling down to the bottom, you get the complete raw sequence, all 966 base pairs. Then, above that, a list of features inside the sequence that the authors thought were important. Here is where we find the synthetic biologist's 3 favorite letters, CDS, a coding sequence for a protein. The authors have indicated that the GFP protein is encoded between bases 26 and 742. You can check this if you want. Beginning at 26, we see a start codon, ATG, and ending at 742, we find a stop codon, TAA. They even give us the exact amino acid translation. Before the CDS, we find the 5' untranslated region. Here's where we expect to see the ribosome binding site, or the RBS. And the sequence also contains some non-coding elements at the 3' end. This is the standard structure for an mRNA, which we expected when the record was described as an mRNA. This GenBank record has given us everything we need for a new biological part. In this case, the part that we want is the CDS. We can use PCR to amplify this sequence from a cDNA library, or we can use gene synthesis to simply order it from a synthesis company. Most of the time, we expect this CDS to work like a biological part. We can add a promoter, an RBS, a terminator, put it on a plasmid, transform it into bacteria, and it will function as intended. In this case, by providing fluorescence to the host bacteria. And that's it. That's how it works. Of course, it's not always quite this easy. GenBank records are very rich with information, and it takes practice and biological knowledge to pull out the functional features and leave behind the junk. And as your parts get more complicated than just GFP, it takes more biological knowledge to make sure that they behave as intended. But the basic process of reading the literature, reading the GenBank record, and reading the sequence is fundamental and common to almost all new SynBio work. GenBank is the place where new parts are born. And there's over 100 billion base pairs of sequence data in GenBank today, so who knows what other cool stuff is in there. So until next time, keep reading the sequences.
Thank you.